The Arrol Adam Fund was set up in memory of William Arrol Adam, who read chemistry at Fitzwilliam Hall from 1901 to 1905. He was a captain and served in World War I on the Western Front, and he was actually injured on 2 occasions. He was shot twice on one, and then in 1918 he went missing and was presumed to be killed, but then reappeared and rejoined his battalion. And he returned to England and lived in Essex until his death in 1940. And the Arrol Adam Fund was established from a bequest made in 1962 from his widow, Jane Adam. And the stated intention of the fund is to disseminate knowledge, promote discussion of issues of general interest and concern, and to foster the plain use of simple English. And with the fund, it supports both this lecture series, the Arrol Adam Lecture Series, and some literary prizes, which we award to students in the college. Tonight's lecture is going to be in 2 parts. We'll first have a talk and then a panel discussion. And I'm going to ask you all to mute your microphones during the talk. But please, if you've got burning questions, use the chat to log those questions, and we will try and come to those during the panel discussion. I now want to just hand over to Dr. Jongmin Choi, who is our Fitzwilliam College Postdoctoral Research Fellow and is in the Department of Plant Sciences, and she's going to introduce our speaker for tonight.
Thank you, John. It is my great pleasure to introduce Giles Oldroyd as today's lecture speaker. Giles received his bachelor's degree from the University of East Anglia in 1994. Then he moved to the States and received his PhD at the University of California, Berkeley. After his graduation, then he did a Howard Hughes Medical Institute postdoctoral fellowship at Stanford University. This is the time that he started to work on symbiosis between plants and microbes, which he's going to talk about today. In 2002, he received BBSRC David Phillips Fellowship and returned to Norwich at the John Innes Center to start his own independent research group. To our great benefit, then Giles decided to move to Cambridge 3 years ago and joined the Sainsbury Laboratory. Last year, he was elected as a Russell Geiger Professor of Crop Science in my home department, the Department of Plant Sciences. At the same time that he became an inaugural director for a brand new Crop Science Center on Huntington Road, which we hope to open in September. This new center aims to advance crop science research by bridging the basic research and then applied crop science knowledge in collaboration with the National Institute of Agricultural Botany. So now let me tell you a few highlights of his many grants and awards that he received throughout his career. Particularly, I'd like to mention one grant that I'm very proud to be a part of. Since in 2012, Giles received a prestigious grant from Bill and Melinda Gates Foundation and has been leading a program called ENSA, which stands for Engineering Nitrogen Fixing Symbiosis for Africa. Under his leadership, the ENSA has grown into a big international collaboration comprised of 70 research scientists from 12 different research groups. And then I'm part of that. So he also received many awards, including EMBO and the European Research Council Young Investigator Award, and also Royal Society Olsen Research Merit Awards. This year, his contribution to science was finally recognized, and he was recently elected as a Fellow of the Royal Society. So please join me in welcoming Giles into this virtual stage.
Great.
There we go, Giles.
Thank you to Jongmin for such a generous introduction. And also thank you to Jonathan for the original invitation. It's a real honor to be presenting the Arrol Adams lecture today and an honor to be talking to the Fitzwilliam Fellowship. I'm really sorry we're not in our beautiful lecture theatre, but I suspect we might even have greater attendance, I don't know, online, or certainly a broader attendance. It looks like a wonderful group of perhaps a lot of Fitz Fellows from years past. So I shall share my screen. Good. I hope everybody can see that. Can I get a thumbs up, Jongmin, if you can see it? Yeah, perfect. Okay, so before I just get to the meat of the science that I want to talk about, I just wanted to introduce what Jongmin already introduced, which is this new center that I'm heading called the Crop Science Center. It's an alliance between the University of Cambridge and NIAB, and it's a really important development for Cambridge because for many hundreds of years, Cambridge has been a center of excellence for fundamental understanding of plants. What the Crop Science Center provides us with is a platform to really start to apply that fundamental understanding to improve the way we grow our food, improving crop plants and improving agriculture. And we have a brand new building that's very close to Fitzwilliam College. It's just down the road, down the Huntington Road. And I'm really positioning the Crop Science Center to be focused primarily on two aspects: sustainability of food production and the equity of food production. And I hope that both of these points, equity and sustainability, you will feature strongly in my talk today, and I hope you get that message. So my research is really focused on how plants acquire nutrients from their environment. So at lunch today, you will probably have eaten a mixture of plant and animal products, and you've digested those products down into amino acids, nucleic acids, polysaccharides, and fatty acids, and we absorb those components into our system, and then use the amino acids to rebuild proteins, use the nucleic acids to build DNA and RNA, polysaccharides to build the complex sugars, fatty acids to build the lipids. But plant nutrition is radically different to that. Plants start from scratch, so they build nucleic acids from scratch and they build amino acids from scratch. So they do so by capturing nutrients in their most elemental form from their surrounding environment. So they're getting carbon from using the energy of the sun through photosynthesis. They're breaking carbon dioxide into the carbon molecules, which they then use to build these biological components, amino acids, nucleotides, sugars, etc. And they're doing that by capturing carbon dioxide from the atmosphere. And the remaining primary nutrients, phosphorus, nitrogen, water, and oxygen, they're capturing these as phosphate, nitrates, and water from the soil through uptake in their roots. Now, the availability of phosphorus and nitrogen in particular can be very limiting to plant productivity. And for this reason, we apply these nutrients at very high concentrations in the form of inorganic fertilizers. And this really massively improves crop productivity. So this was the foundation of the Green Revolution that led to a tripling, more than a tripling of our cereal production. So maize and rice and wheat are highly dependent on the application of nitrogen and phosphorus. And now with the scale of our global population, we're really fully dependent on the productivity that this system provides. But the system is actually very inefficient. I would argue that if this was a industrial system, we would not accept the inefficiencies that we accept in food production, because we apply these nutrients, but actually only a minority of those nutrients get into the crop plants. A lot is lost out into the environment, with an awful lot of nutrients being lost through agricultural runoff, where they get into our aquatic systems and cause a process called eutrophication that really leads in a collapse in the biological diversity of our aquatic systems. But these nutrients are also having impacts in our terrestrial systems as well. And these impacts on biodiversity are happening across the planet wherever we're growing food with the use of nitrogenous fertilizers. And in fact, the image on the bottom here from Australia was just from the last growing season last year, where they had this awful die-off of fish die-off in the Murray-Darling River because of these agricultural nutrients in their environment. We also lose these nutrients through a second process, a process called denitrification, which is the action of bacteria in the soil that leads to the volatilization of nitrogen as nitric and nitrous oxides. And these escape into the atmosphere where they're extremely potent greenhouse gases, 35 times more potent than carbon dioxide. So the use of these inorganic fertilizers is driving down biodiversity through their release into the environment and driving up climate change through the volatilization of reactive nitrogen molecules. And in fact, the efficiency of the system is extraordinarily poor. We're only capturing globally about 33% of applied nitrogen, and even worse for phosphorus, about 16% of applied phosphorus. So globally, around the globe, the vast majority of the nutrients that we're adding to the system are actually escaping out into the environment, having these pretty unpleasant consequences. Now, the inorganic fertilizers started to be used in the 1960s with the advent of the Green Revolution, and their use has really tracked population growth. So up to now, with a population of 7 billion on the planet, we've already doubled the global nitrogen cycle. So we've increased the amount of reactive nitrogen on the planet by twofold, by the current use of inorganic fertilizers. And in this graph coming from the UN, you can see that we predict that fertilizer use simply tracks population use. And in this business as usual scenario, we'll see another doubling of the global nitrogen cycle. So in all told, we'll see a quadrupling of the amount of reactive nitrogen in the environment. This is really not sustainable, and that will have really profound impacts in the biodiversity in our food production areas. Now, when you look across the planet at who's utilizing that nitrogen, it's really the rich world nations, North America, Europe, North India, China, heavy users of nitrogen as fertilizers. And these are the countries where just really experience the massive problems in eutrophication in their aquatic environments. In contrast, farmers in sub-Saharan Africa, you can see their use of nitrogen is very, very limited. And that's because these farmers are smallholder farmers with very little financial resources, and they simply can't afford the nitrogen fertilizers. And as a result, their crop productivity is very poor. So we're looking in the average smallholder farmer, they're producing somewhere in the range of 10 to 15% of their potential productivity, in contrast to a rich world farmer, which is achieving about 90% of potential productivity. So I would argue that the access to and the application of nitrogen and phosphorus really sits at the heart of equity and sustainability in agriculture. And I personally think that we have a broken system, and it's certainly not a system that we want to rely on when we get to a population size of 10 billion people, because the environmental implications are pretty scary. So in my research and the program that I lead, funded by the Bill and Melinda Gates Foundation, we're striving to change this system and to replace as much as possible the inorganic fertilizers in agriculture. So it's not just in agriculture where fertilizers are, where the nitrogen and phosphorus is limiting. It's also limiting in natural ecosystems. And in natural ecosystems, plants turn to microorganisms to help in the uptake of these limiting nutrients. And so there's a number of critical microbial associations that plants engage with. And these are beneficial microbial associations where the plant benefits from interacting with these microorganisms. And there's the two primary ones that I'm going to talk about today is the arbuscular mycorrhizal association, which is an interaction between plant roots and a group of fungi. And in these association, the fungus heavily colonizes the root. You can see on the right here is an image of a root, and all this gray staining is the fungal hyphae that's colonizing the root. And they form these highly branched structures called arbuscules, which are growing inside a plant cell. So just where my arrow is a point where the fungal hyphae goes from growing between the cells of the root to growing inside a cell of a root. And inside the cell of the root, it forms this incredibly highly branched structure. And around all of that branched fungal hyphae is plant membrane. So they create an enormous surface area for the exchange of nutrients. In addition to colonizing the plant root, the fungus also colonizes out into the soil, as shown by this diagram. All this sort of web-like structure is the fungal hyphae growing out into the soil. And it makes a much, much larger surface area for the capture of nitrogen, phosphorus, as well as water. And then the fungus is capturing these nutrients from the soil and then transmitting them into the internal hyphae and eventually releasing them into the plant cell through these intracellular arbuscules. So the arbuscular mycorrhizal symbiosis is helping in the general uptake of nutrients from the soil. But the second symbiosis I'm going to talk about is a really radical association that occurs between some groups of plants and a group of bacteria that can fix nitrogen. Now, nitrogen itself is not a rare element. It's a very prevalent element, but its primary form is as molecular dinitrogen, which is in the atmosphere. So 80% of the air you're currently breathing is molecular dinitrogen. This is a very unreactive form of nitrogen that very few organisms can use. And the only organisms on the planet that can use molecular dinitrogen are bacteria that have an enzyme that can break the triple bond in this molecular dinitrogen and convert it to ammonium. And ammonium is now a reactive form of nitrogen that many organisms can use, in particular plants, can use ammonium to incorporate in and build the amino acids and nucleic acids that are essential for proteins and DNA, etc. Now, in this association, we see quite complex developmental changes. We see the formation of these nodules on the roots of the plant, these pink structures, which are unique structures that are formed to accommodate these bacteria. And the plant also forms these tubular infections. So what we're seeing here is a root hair cell that grows out of the surface of the root that would grow normally into the soil. And these root hairs can find the bacteria in the soil. They trap the bacteria by growing around the bacteria, and then they form a tubular infection into the cell in which the bacteria grow. So the bacteria are staining blue inside that tubular infection. At the same time, the plant initiates the formation of these nodules, which you can see on this cross section. We get a lot of cell division forming, and this occurs alongside these tubular infections that grow down into the cell. Here we're seeing a cross section of one of these tubular infections, and these grow all the way into the dividing cells of the nodule, where they release the bacteria into, you can see these bacteria blebbing off this infection threads, and they're released inside the cells of the nodule in these membrane-bound compartments. And then ultimately you see here a cross section of a nodule, and on the right is an infected cell. You can see massively infected by bacteria, always surrounded by plant membrane, in contrast to an uninfected cell in the nodule. So this ends up being a massive infestation of the plant root tissue with these nitrogen-fixing bacteria, with the bacteria delivering all of the plant's nitrogen needs, coming from the fixation of molecular dinitrogen from the atmosphere. So in the natural ecosystems, plants can utilize these associations with fungi and with bacteria to help in the uptake of these nutrients. In my research and in the program that I lead, we're striving to utilize these microbial associations to replace nitrogenous fertilizers, or replace as much as possible nitrogenous fertilizers. Now, the arbuscular mycorrhizal association, the fungal symbiosis, evolved very early in the land plants. And what I'm showing you here is the phylogeny of the totality of the plant kingdom. So green algae were the precursors to plants, and then the very primitive plants like mosses and liverworts emerged first in the plant kingdom. Then we have lycopods, ferns, gymnosperms, which is your conifers, and then we have this huge group of the angiosperms, which are all of our flowering plants, that are split up between monocots, which includes the grasses where all our cereal crops sit, and then the bigger group of the angiosperms, the dicotyledonous angiosperms. Now, the arbuscular mycorrhizal symbiosis, we know, evolved very early in the evolution of plants. It sits right at the base of the land plants, and as a result, it's ubiquitous across the plant kingdom. So most species of plants can form this fungal association, including our cereal crops like wheat, maize, and rice. So here, a maize plant I'm showing you has the ability to form the arbuscular mycorrhizal symbiosis, but unfortunately, plants are very intelligent, or fortunately plants are very intelligent, but in this case it's rather unfortunate, because when we apply the nitrogenous fertilizers and have high levels of nitrogen and phosphorus in our fields, the plants just recognize there's lots of nitrogen and phosphorus around, and they don't bother entering the symbiosis with the fungus. Instead, they just take the nitrogen and phosphorus up directly from the roots. Now that's slightly unfortunate, because as I said, the fungus is actually much better at capturing these nutrients, which would mean much less loss of the nutrients to the environment and more capture into the plant. So an optimal situation would be this, where we have the mycorrhizal symbiosis even when there's high concentrations of nitrogen and phosphorus in the situation when you fertilize the crop plants. And at least in the lab, through a foundational understanding of how these systems works, we've been able to rewire the system such that we can drive that high fungal colonization even when there's ample fertilization. So here I'm showing you some data that we've generated pretty recently within the project. And what I'm showing you here are graphs that measure the total colonization of the root by the fungus. So on the x-axis is the percentage colonization, and under low phosphate conditions, the root is colonized up to about 75% of the root is colonized by the fungal hyphae. In contrast, when we go to a high phosphorus condition, we switch now to much lower colonization. So you see we go from 75% colonization under low phosphate down to 25% colonization at this concentration of phosphate. But here we have some engineered lines as well, where we've misexpressed some transcription factors that control that nutrient suppression of mycorrhization. And when we misexpress these transcription factors, so we're just driving their expression constitutively in the root, we now can have mycorrhizal colonization at very high levels, even at these high phosphate concentrations. So this line that's overexpressing one of these transcription factors, you can see that even though it's grown under high phosphate conditions, it's getting colonized almost to the same level as the wild type at low phosphate conditions, even though this plant is grown under the equivalent conditions as this wild type here, the high phosphate. So at least in the lab, we've overridden this nutrient suppression of the fungal association. So theoretically, we've already achieved, at least in the lab, this. Now, those results are just very fresh. We got them first last summer. We've just repeated them again this year, so we know it's all correct. And theoretically now we have plants that where we can drive the mycorrhizal symbiosis in agriculture, which theoretically we would presume would massively improve the uptake efficiency. But to really demonstrate that, we need to get those crops into the field, and that's really where the Crop Science Center comes into play, because it gives us the platforms to put these engineered crops in the field and test to the degree to which they improve the sustainability of the farming practices, driving improved uptakes of nitrogen and phosphorus, and hopefully means that we can apply less nitrogen and phosphorus and still get the same or equivalent yields. And I'm just reiterating the point, we've got a long way to go to improve the efficiency of capture of these nutrients. So if we can double the amount of nitrogen that's captured through these mycorrhizal symbioses, we can really massively reduce the amount of nitrogen going out into the environment. So for me, that's phase one of reducing our reliance on these inorganic fertilizers. And at least there, we know in the lab we've achieved that. We're now getting out into the field to see how effective those crops are. The second phase of this program is really to just remove the need for nitrogen in totality. And that is utilizing these processes that have already evolved in the plant kingdom that have utilized the nitrogen fixing bacteria instead of nitrogenous fertilizers to deliver the plant's nitrogen needs. Now, in the evolution of plants, this nitrogen fixing symbiosis evolved much later than the fungal association. So the fungal association is sitting right at the base of the land plants about 450 million years ago. In contrast, this nitrogen fixing bacterial symbiosis evolved about 100 million years ago, and it's restricted to a group of the dicotyledonous crop plants that include the legumes, like peas and beans. Unfortunately, it didn't occur in the monocotyledonous crop plants where our cereals are, the maize, rice, and wheat. So maize, rice, and wheat can't form the nitrogen-fixing symbiosis. So essentially in the project, through an understanding of what happened at this point in evolution, we can then transfer those evolutionary steps into our monocotyledonous crops so that we can allow monocots to benefit from this bacterial nitrogen-fixing symbiosis. Now, this is really where all that foundational knowledge that's been going on in the academic sector comes into play. So I spent 20 years of my life trying to understand how these associations form. And it's not just my research, but it's many other research groups around the world, has given us a really strong understanding of how legumes, peas and beans, form this nitrogen-fixing symbiosis. And this has given rise to what we're drawing here, which is this signal transduction pathway that is present in plant cells that allows them to recognize this signaling molecule that's coming from the nitrogen-fixing bacteria. Now, at the core of this signaling pathway are oscillations in calcium, and this is a process by which plants are transmitting the information from recognition of bacteria in the soil to the gene expression changes that occur in the nucleus. And in this movie, you can see these oscillations of calcium occurring in the nuclei of cells on the surface of a root. And these cells will pulse their calcium continuously in response to the addition of this signaling molecule from the bacteria. And this is essentially the signal transduction. What you see in, it's a bit speeded up, but what you see happening is essentially the cells telling themselves that they've seen the nitrogen-fixing bacteria out in the soil, and then are activating the signal transduction pathway that is then, that calcium signal is transmitted to a bunch of proteins that ultimately drive gene expression changes in the root that allow the root cells to now accommodate those nitrogen-fixing bacteria. Now, if I had to, from an engineering perspective, transfer, even though we know a lot about this signaling pathway, if I had to transfer this, engineer this whole signaling pathway, and in particular those oscillations in calcium, into a cereal crop, that's a pretty daunting undertaking. But thankfully, because of the history of evolution, that signaling pathway didn't actually evolve for the nitrogen-fixing symbiosis. It actually evolved much earlier for that arbuscular mycorrhizal fungal association. So our cereal crops already have this signaling pathway, and I've really simplified that down to just a very simple view of that signaling pathway. And in our cereal crops, that signaling pathways are now allowing just the recognition of the fungus, and the signaling pathway activates a process of a number of proteins that we call the infectosome that allows that intracellular infection by the fungus. So this framework is already there in our cereal crops because it evolved for the fungal symbiosis. Now, in the infectosome that the symbiosis signaling pathway is controlling right here, that allows the fungal symbiosis, is also playing a role in the bacterial infection. And what that infectosome is a protein complex that sits at the tip of those infection threads. These are these tubular invaginations that allow the bacteria, shown here in the purple fluorescence, to intrude into the plant cell and grow inside the plant cell. It's the infectosome that guides that intrusion and bacterial intrusion into the plant cell. And that infectosome is already there in the plants. So the framework that was already evolved to support the fungal symbiosis was adapted in legumes to now allow the bacterial symbiosis. So we have this core framework here that now is utilized to give bacterial infection. And then that framework was hooked up also to developmental changes in the root cortex to give rise to the nodule. Now, from an engineering perspective, the core framework is already there because it's already there in our cereal crops because it's there for the fungal symbiosis. And we also know that the developmental processes that drive the formation of a nodule are also there in our cereal crops. So from an engineering perspective, what we're focusing on is the wiring, it's the red parts. It's really just the network, how you network the signaling pathway to the developmental changes in the root, as opposed to having to engineer the totality of these processes. And from an engineering perspective, that's a much simpler thing to engineer the connectivity of these processes than to engineer the signaling pathway and the developmental processes in totality. So from an engineering perspective, this is what we're focusing on, is connecting up these existing networks that already exist in cereal crops from an understanding of what we know in legumes, to allow the nitrogen fixing symbiosis. And just to show you that really we can generate nodules, these are pseudonodules in barley in a cereal crop. The developmental processes are already there in cereal crops to make nodules. It's just that the wiring is not there to link that developmental processes to the recognition of nitrogen fixing bacteria. Now, it's still quite a daunting exercise to do all of this. It remains aspirational. This is what we're really focusing on in the Bill and Melinda Gates funded program, is to see if we can transfer this nitrogen fixing symbiosis. But I think that the key message I want you to get across is that there is already a framework in our cereal crops that would theoretically makes the engineering much, much simpler. So this is where I'm aspiring to be. I want to essentially get rid of nitrogenous fertilizers as much as possible by transferring the nitrogen fixing symbiosis from legumes to cereals, allowing our cereal crops to get their nitrogen from the atmosphere, which is a far, far more efficient and sustainable way of getting nitrogen into agriculture, and then to minimize the amount of phosphate that we apply in agriculture by bringing in also the fungal symbiosis into our agricultural associations. And if we can drive all of these microbial associations in our agricultural soils, we'll have a far more sustainable way of producing our food. But it's also a much more equitable way of producing our food, because for a smallholder farmer, they no longer would need to buy nitrogen because the plant can just get its nitrogen from the air, and they'd need to apply much lower levels of phosphorus, making it much cheaper to grow their crops. Now, the impacts for smallholder farmers could be extremely profound. So I'm showing you here the productivity of maize in response to nitrogen. And smallholder farmers are applying extraordinarily low levels of nitrogen currently. So by just bringing in the nitrogen symbiosis, we're looking at potentially tripling their yields or even quadrupling their yields, depending on the farmers that we're talking about. But to impact out the first world farmers in the developed world, we have a problem, because these symbiotic associations don't come for free. The plant has to pay with carbon for the nitrogen and phosphorus benefits that come from symbiosis. So there's a bit of a yield penalty if you compare symbiotic nitrogen and phosphorus growth rather than nitrogen and fertilizer application alone. And this yield penalty is going to be problematic for first world farmers, because they just want to keep growing the same amount of food. They don't want to see a reduction in food production, even though we can drive the sustainability. So it's the big problem for a developed world farmer is that if you rely on these microorganisms, if you rely on these microorganisms to deliver nitrogen and phosphorus, you have to pay for that with carbon from photosynthesis. And this is, for me, where it really gets very exciting working with the Bill and Melinda Gates Foundation, because we're all focusing down here on driving, improving the sustainability and equity of nitrogen and phosphorus delivery. But parallel programs also funded by the Bill and Melinda Gates Foundation and the UK Department for International Development are also looking at increasing the efficiency of photosynthesis. And this is a program led out of the University of Illinois in the US. And there, they've already increased the efficiency of photosynthesis. So it gives you more carbon capture in the plants by just driving up the efficiency of photosynthesis. Now, in their models, they predict they can increase the efficiency of photosynthesis by 130%, but they've already increased that efficiency by 40%. Now, I know that the amount of carbon, extra carbon I need for all of my microbial services is about 40% more carbon. And the Wright project has already got that from increased photosynthetic efficiency. So if we bundle the increased photosynthetic efficiency along with the symbiotic nutrient uptakes, then we get to the point where we no longer have this yield penalty because we get more carbon in from photosynthesis. So we lose this yield penalty now, so we get even better productivity for smallholder farmers, but we can also drive the sustainability of first world farming systems. And for me, this is the vision of a really, truly transformative agricultural system. Because if we can achieve all of this, which is where we're trying to go in the Bill and Melinda Gates Foundation programs, then we have these amazing crops that give us even greater productivity through the increased photosynthetic efficiency without all the pollution derived from nitrogenous fertilizers. And if we can get to this, and we really address global food security, but global food security in a radically new world of sustainability. And I think that for all the challenges we face going forwards for 2050, with an expanding population and all the problems that we have with climate change, we really need to find a better way of growing our food. And for me, I think this is the optimal way where we can drive up productivity and drive up sustainability and drive up productivity for smallholder farmers using these technologies. So with that, I'd just like to acknowledge my fantastic research group that I work with, and then also the whole program, the Engineering the Nitrogen Symbiosis program, which is a European-led program, primarily countries across Europe. These are all of my collaborators in Europe. And in particular, it's a real pleasure to work with the Bill and Melinda Gates Foundation. It opens up the potential for impact, as well as the Department for International Development. And I'm also supported by the Gatsby Charitable Foundation, which is the David Sainsbury's Charitable Foundation, as well as our Biosciences, BBSRC, our UK research organization. And with that, I'll be happy to take questions, but in the context of a panel discussion.
Thank you so much, Giles. That was fascinating and hopefully very understandable as well. We're going to move now to a panel discussion, and we intentionally tried to bring into this panel discussion people from Fitzwilliam, from the Fitzwilliam community, but at different levels of their academic careers. So we've got Chai Hao Chu, who is a PhD researcher in plant sciences. We've got Jungmin Choi, who you've already had introduced Giles's talk, who's a postdoctoral researcher in plant sciences. And we've got Professor David Coombs, who's the head of the Forest Ecology and Conservation Group and the director of the University of Cambridge Conservation Research Institute. And Giles, of course, in the panel. And we're going to start by just letting the other three panelists reflect on or ask some questions of Giles. So I'll hand over now to whoever wants to go first.
Jungmin.
Thank you, Giles, for wonderful talks. This motivate me more to do more work, actually. So I was wondering, what do you think is the bottleneck at the moment that translate this basic science into the field? That
I think, it's probably the biggest bottleneck we struggle with is plant transformation. So that is introducing new genetic material into plants. It's a very slow and laborious process and limits the amount of what we can do. And that's just always going to be a challenge. But we can increase the throughput of plant transformation, but we're probably not going to be able to improve massively on the efficiency of plant transformation. But I think I would say that's probably the primary bottleneck. I think it's all possible. That bigger vision is certainly possible, but it's just it's a slow turn of the wheel when we're working in crop plants and introducing new genetic material into crop plants.
Thank you.
David.
Well, thank you first, Giles, for your talk. I liked the reference John made at the start to the Arrol Adam Fund being about fostering the use of plain and simple English, and it's remarkably hard to do that in the biosciences with all those complicated molecular pathways to think about. And I thought you did it brilliantly, so thanks for that. I wanted to reflect a bit on the consequences of these technologies if they were to happen in the future on the twin crises we face, the climate crisis and the biodiversity crisis. And You mentioned a bit about the sort of changes in yield which might be anticipated. We're going to reduce the amount of fertilizer we need to put on the fields if these crops are engineered with the mycorrhizal and nitrogen fixing bacteria. But that might have consequences for yield, and then we'd have to expand even more into the natural world, perhaps. And I just wondered whether people have thought about these sort of potential unintended consequences in this broader context.
Yeah. So unintended consequences. It's always an interesting challenge there. You know, I mean, we're putting something radical into cereal crops, making them able to fix nitrogen, well, we already eat legume crops. I mean, we're just taking genes from legumes and putting them into cereals. So we're taking it from crops that we already eat and putting them into another crop that we already eat. So from a perspective of, I mean, of course, you have to do all of the appropriate health and safety measures to make sure that we are that the new crops are safe. But we are already eating these genes when you eat peas and beans. So I think the bigger consequence is broadening the plant capability to fix nitrogen, and that's certainly something we have to consider. And we would do so by a lot of trial and error in the field before we were to release those crops. But I would counter that. You know, the tendency is to say these technologies sound a bit scary, and what you're doing could sound a bit scary and radical. So you're scared about the unintended consequences. But if we don't do anything, we just fill the planet with reactive nitrogen with very intended consequences. We know what that's doing. It's driving massive biodiversity collapse of our aquatic systems and declines in biodiversity in our terrestrial systems. And if you were to quadruple the global nitrogen cycle, which if we do nothing, that's what's going to happen. With the population is growing, people want more meat. The only way that's going to happen, the only way we're going to achieve that is more fertilizers. I don't want to live on a planet with 4 times more reactive nitrogen. It's going to be a lot of grass and a lot of green rivers and a lot of green ponds. And we're also, the consequences on our oceans and seas, you know, we're seeing eutrophication now. A report just recently showing that eutrophication has expanded massively in all of our seas surrounding agricultural areas. But if we quadruple the reactive nitrogens, the amount of the nitrogen cycle, we're going to have profound implications on our oceans, which we haven't even yet seen. You know, eutrophication in the oceans, it's really around our seas, but we're already seeing nitrogen levels going up, and that's going to, if you have eutrophication in our deeper oceans, that's really bad news. So I think, you know, for the climate consequences and our biodiversity consequences, we know what that is, and I don't think that that's a sustainable or a future way, and I don't see another solution. I don't see how you feed 10 billion people. I don't believe we can do it with purely an organic farming system, because the yield penalties for organic are too severe. Well, we could all become vegetarian. That's possible. But I just don't see that, certainly not happening. And it's not the direction of travel. And I don't see many governments enforcing vegetarianism on their population. So unless we have a radical shift, and we have to talk a global shift, not just here in the UK, but you need China, 1.2 billion people in China being vegetarian. I just don't think this is going to happen. So I think that we have to turn to technology to solve these problems. I don't see another way, personally. Jongmin, we do have a lot of questions on the chat. I hope you are seeing this.
Did Chai have a question though first? I was going to see.
Yeah, I have a question. So in the case of benefiting the smallholder farmers, so instead of having them not having to purchase nitrogen fertilizers, then when they actually end up having to work with the genetically modified seeds, how would that be provided to them?
I'm sorry, can you just repeat the question, Chai? I didn't.
So in the case of a more equitable, more equitable farming system where smallholder farmers can get more equitable access to inputs to production.
Yeah, I understand. Okay, I understand what you're saying. So right now, the big challenge is that smallholders just can't afford fertilizers, and we don't have the infrastructure to get fertilizers to them. In these, if we get nitrogen-fixing cereals, the technology sits in the seed, and seed is much easier to get to farmers than fertilizers is, and farmers can share seed. Now, all the technologies that I'm describing will be owned by, in the first instance, University of Cambridge, but because of our licensing agreements with the Bill and Melinda Gates Foundation, they ultimately get licensed to the Bill and Melinda Gates Foundation. So they're the ones who end up owning the technologies. And their mission in the world is to benefit the poorest farmers on the planet, or the poorest people on the planet. So it's actually very exciting working with the Bill and Melinda Gates Foundation, that the technologies we're working on will be to the benefit of the foundation. And the foundation is just putting together a new entity called Bill and Melinda Gates Agricultural Innovations, which is striving to produce the products that I've just described and get them into the crops that smallholder farmers grow. This is a radical change in the way that food is currently, or food crops are currently produced, where we're heavily reliant on a few big biotech like Monsanto, which is now BASF. But there you're looking at a for-profit model that aims to make profit from farmers. This is not the model that the Gates Foundation is striving to use. They're looking to improve the livelihoods of smallholder farmers, and we're developing these technologies for the benefit of smallholder farmers and getting it working in crops and getting those crops to smallholder farmers. So I feel like we're trying to transform the way we grow food, but also transform the way we make those food crops in a way that would benefit smallholder farmers rather than a for-profit model of the current big biotech.
Thanks, Giles. Just on that note, there's a question here from Natasha Grist asking, what's the time frame that that's a hard question for you because it's research, but
yes.
When do you think the smallholders will get it?
Well, that first technology I talked about, which is really driving the fungal association into our arable farming systems, we have it working in the lab. So now we will have 3 years of field trials in the field to see the implications of that technology. If it's working, then if it works within the first years of the field trials, we're starting to develop it as a product. In that case, we're looking at, even so, product development takes time. We're looking at sort of probably a 10-year window to have that as a product in farmers' fields, because there's still quite a bit of optimization, and we hope it works. So we still have to get it into the field. For engineering nitrogen fixation, that's a much harder time frame to put on. But I would just say that I've been running my research group, or I've been in the field of plant molecular genetics for 25 years since I entered the lab, my first lab, as an undergraduate researcher. And back then, we didn't even know what the genes were inside plants. And now we have this massive transformation because of all the technological advancements. In 25 years, we've transformed our understanding of how plants work. And we're really seeing a revolution in biology and a revolution in our understanding. And I think the next 25 years, we're going to see a massive expansion in that revolution, in the way we can use all that biological understanding to see transformation, not just in agriculture, but also in medicine. I really believe that in the next 25 years, we will see a transformation in what we can do in biology. And I hope I've got 25 years left of my career. So I hope I've got long enough to see it out and to see it to nitrogen fixing cereals, crops in the field. And I think that at the moment, I think that's a realistic time frame. But, we are trying to create something that doesn't exist. So we don't really know how long it's going to take.
Mm-hmm. Did any of the other panelists have anything to comment on timeframes? No, you're good. Okay, we've got some questions here. So there was a clarification question about the meaning of misexpress. And I guess that's from Simon, and also Rachel Webb asked about unintended consequences of these interventions in terms of Mother Nature. And so you've touched on that a little bit, but Do you want to?
Yeah, so I can start with misexpression. So what we're doing with those transcription factors is they're transcription factors that already exist in cereals, but what we've done is rather than these proteins are regulated by promoter regions, which are linked to the nutrient status. So the transcription factors are switched off when there's lots of nutrients around, and we've just changed the promoter so those transcription factors are no longer suppressed by that nutrient status. So now they're expressed in a differential context. So it's the same transcription factors are already there in the cereals, but they're expressed in a different context because we changed the promoter region of the gene. We call that misexpression. You can do it with genetic engineering, but you can also do it with gene editing, which is a much more focused technology that doesn't leave transgenic DNA. So I think we can do it without ending up with a genetically modified crop. But for nitrogen fixing cereals, we can't do it without having a genetically modified crop. So I'd be interested in a sort of broader discussion on the panel because I get this frequently, unintended consequences. And I think that that's too often used just to stop technology from moving forwards. We sort of get scared of these technologies. So we run away. We sort of say you shouldn't do them. But, currently, if you're a diabetic, you are injecting yourselves with genetically modified insulin, genetically modified protein injected directly into your bloodstream. Now, we're talking about food where you digest it down to the amino acids and then you just absorb those amino acids. So by the time you actually digested the protein, your bloodstream doesn't even see it. So I feel like there's very different standards are given to medical research with regard to GM versus agricultural research with regard to GM. And there's an enormous health and safety procedures that you go through to make sure your food is safe. So I don't think there's any issues with regard to food safety. And what we're talking about is taking genes from peas and beans that you already eat and putting them into wheat and barley and maize, which you eat. So we're not talking about radical changes in the genetic material that you would actually consume. So I think the primary unintended consequence or intended consequence is the implications of moving the capability to fix nitrogen to something broader across the plant kingdom. That is quite a radical. I fully acknowledge that's a radical shift. But I go back to my original statement. If we don't do this, then we're just going to fill the planet with reactive nitrogen. And we know the consequences of that, and it's not good. So we drive up climate change, we drive down biodiversity. And I think if we just say, okay, you can't do these technologies because we're scared about potential implications, it's not as if the alternative is a business as usual scenario, which is just a lot more fertilizer.
Yeah, the unintended consequences of the Haber-Bosch process are already in society. And yeah, and David, do you want to comment on that at all?
Well, I completely agree that about the Haber-Bosch process, and not only affecting aquatic systems, but a lot of woodlands and grasslands in the UK. Natural woodlands and grasslands are polluted with these industrial nitrogen fertilizers as well. So it's not just the aquatic systems. And at the world level, in the future, we need to feed even more people and bring people out of poverty who are currently too little to eat, so we actually have to produce more food. So somehow increasing productivity on the agricultural land, trying not to push into the remaining bits of nature if we can, is the game. And so we have to actually increase productivity on our agricultural systems. So we can't go to organic systems, which we know will reduce productivity. So I think this is a very interesting way forward. And my question wasn't intended to be talking about the genetic engineering, but the other potential unintended consequences that Giles was alluding to there.
Thank you. Probably because we're very close to finishing it at 6:30, but just one more question. We had a couple of questions, one from Samantha Islam and one from Eleanor Wilding, that talk about this technology and how it might be affected by future climate change or changes in temperature, moisture, acidity, etc. that we face. Is it future-proofed, I guess, is the question, Giles?
So I think that when you look at climate change, you know, we're talking about more extreme climatic events. And so you often see in Africa, like what's happening in Zambia right now, they're going into a severe drought. And the farmers, in previous years, their productivity has been 15% of the potential productivity. So they're hand to mouth continuously, barely hand to mouth, you know, and then you have a drought year and they have nothing. And they have no fallback position. They've got no financial fallback position. They don't have any food, so they go into starvation. So I think actually the best way to address climate change is to maximize productivity for these farmers. If they were producing 80 to 90% of their potential productivity year on year on year, then you have a financial buffer when you have a drought. So even, you know, you have a drought, your productivity falls down, but you don't then end up with mass starvation because you have farmers who are producing enough that they can cope with a few years of poor production because they have a fallback position to go on to. So I think that the best way to future-proof agriculture is to maximize productivity when the going is good. And, you know, at the moment, for smallholder farmers, their crops are growing to the nitrogen potential. They're not growing to the water potential. When it rains on their crops, they don't grow to the amount of water that's there. They grow to the amount of nitrogen. So if you remove that bottleneck of nitrogen, when it rains, they grow to the water potential instead of the nitrogen potential. And I think that that is really the best way. You maximize the use of the water when it's there by removing the bottleneck of phosphorus and nitrogen. And then you maximize the productivity when the years are good, which then gives the farmers a buffer for when the years are bad. And that's just, I mean, that's how the system works in a rich world nation. And we simply have to create that capability for poor farmers as well. I mean, of course, with climate change, you need to look at other things, heat stress and drought stress. But if you're just producing 15% in a good year, then you're always going to be stuffed when it's a bad year.
Thank you. That's great. I think we need to close out now. So we're out of time. But just to say, I hope you enjoyed tonight's Arrol Adam lecture. And I want to thank, firstly, Giles for being our speaker, which was great. And our panelists, which was Chai and Jungmin and David. Thank you very much. And also just for Fitzwilliam College and the staff behind the scene who do lots of work to set these up. So Alison, Nick and Nicola in particular. Thank you. We hope to do more of these types of lectures online. So we hope you will come to the next one. Bye.